Well, hello, class. Today, we're going to go over the genetic code.
Well, whoopsie freaking do!
Oh, come on, it's fun. We love the genetic. So let's start out with a little review of molecular genetics between the procs and the eucs. Ooh, that'll make you puke, sorry. Right? Molecular genetics, stuff that happens to genes on a molecular level. All right, so in the procs, we have DNA going to RNA. What do we call that? Transcription, our good friend transcription. With a C, DNA to RNA. What about RNA to protein, right? Polypeptide, also known as protein. RNA to protein, that's known as translation. And as you can see here in this picture, in a prokaryote, this is all within one cell. Prokaryotes do not have organelles. So transcription and translation all happen in the same place. Because they all happen in the same place, they can all happen at the same time. As soon as enough of this piece of RNA comes off from transcription, ribosomes can hop on it immediately and start translation. So just like multiple RNA polymerases can sit on a gene to do transcription, multiple ribosomes can sit on an RNA to do translation. How is that different from the eukaryotes, you might ask? Well, let's just take a looky-look. Here we see the gray is the nucleus. There's our little double membrane. Within the nucleus is where we have transcription, right? Transcription happens here to make RNA. Pre-RNA, primary transcript RNA, we have to process it to get mRNA, right? We must 5' cap and poly A tail on the 3' end and splice out any introns. From there, we have to come out of the nucleus to go to the cytoplasm where the ribosomes are. And now a ribosome, our good little friend there, right, hops on to the RNA and reads from 5' to 3'. Again, multiple ribosomes can hop on a eukaryotic mRNA just like they can in the prokaryotic mRNA. Okay, so about the genetic code. But first, a little bit of review, remind us, we're heading into translation, right? We need to understand the genetic code or how mRNA is read in order to turn, quote unquote, or use mRNA as our template for protein. So here's our polypeptide chain or our protein, right? And we have a bunch of sequences of nucleotides that then have to become a sequence of what? What makes proteins? Yes, amino acids. When we went from DNA to RNA, it was a 1-to-1. For every nucleotide in DNA, resulted in one nucleotide in RNA, right? It was, oh crap, I messed it up already. No, stop it, stop. Oh my God. Okay. Sorry, had a moment. Right, 1-to-1. Every nucleotide in DNA is complementary base pair to one nucleotide in RNA. We know now that 3 nucleotides in an RNA equals 1 amino acid, right? 3 ribonucleotides together equals 1 amino acid. Well, how the heck does that work? Well, that's what we're going to find out in this little lecture. Okay, let's remember. What the hell is this? What is this chemical formula for? Yes, it's an amino acid. Okay, remember there's always an amino group with the nitrogen. There's always a carboxyl group, right? COO negative or COOH, right? And then our side chain. Remember you guys had to know all of these in 107 and you'll have to remember the structures again for 380, not for 360, but you need to understand this is the N-terminal. Oops, oh my God, terminal, right? This is the C-terminal or C-terminus, C-term, N-term. the beginning, the end, right? Analogous to 5' and 3' for polarity, but for amino acids or for proteins, we call N-terminus and C-terminus as their polarity. The N-terminus was always what's flapping off on the first amino acid that was added to the chain. The last amino acid added to the chain always has its C-terminus flapping off. And remember, what is this reaction when you put one amino acid and another amino acid together? Yes, it's a peptide bond, and it forms through a condensation reaction. Okay, so we lose water, condensation. That's called a peptide bond. Peptidyltransferase actually makes that happen, which we'll talk to in a minute. When we get to translation, a little fast forward, it's the ribosome, the large ribosomal subunit that has that enzymatic activity that creates that peptide bond. Flash forward. And then remember, the sequence of amino acids is the primary structure, and then secondary structure is how it folds up, mostly because of the backbone, the interactions, the hydrogen bonding in the backbone. Tertiary structure is all the bonding in the R groups, and then the fourth level is when more than one polypeptide chain, more than one individual protein gets together to actually form a functional molecule. So don't forget that about proteins. Okay, so trying to figure out how the genetic code worked was a series of experiments, of course, because we don't just know things, we have to do experiments. And so it makes sense that since there's 20 amino acids total, it cannot be 1-to-1, right? There's only 4 nucleotides. That means there's only, if it were 1-to-1, only 4 amino acids would actually be in proteins, or each of the 4 nucleotides would have to somehow code for 5 different amino acids. That's not going to happen. That didn't happen. That's not how it works. If only 2 letters specified each amino acid, we'd only have 16 still, and there'd still be some redundancy, or some amino acids wouldn't work, and neither one of those are true. So to accommodate all 20, we need at least 3 letters, a triplet code, and that would give us 64 different options. So that's too many, but at least it's enough. So that's our best option. And so we did some experiments to figure out how that works. Okay, so what we see here, if there's potentially 24 codons based on this triplet code, and only 20 amino acids, and if each amino acid only had 1 codon, we'd have all these codons left over that wouldn't code for any amino acid. When we look at RNAs that are functional, there are all 64 codons represented in those mRNAs. And they don't skip any, so they must code for something. So we can't, it's not that we just use 20 of the 64 codons, we do use all of the codons. So what does that mean? That means more than 1 codon must code for each amino acid, right? Each amino acid can be coded for by more than 1 codon, which means the genetic code is degenerate, right? Each codon individually, each one of these 64 only codes for one thing, right? But there's more than 1 codon for each amino acid. Right, so here's some other rules or some characteristics of the genetic code. Yes, it's triplet. Yes, 3 nucleotides. Comma-free, it just means it doesn't skip any bases. It reads 3, 3, 3, 3. It doesn't go, ooh, there's a nucleotide, but I'm not going to read it because I don't like it. It's ugly, and move on. No, it reads 3 by 3 by 3. Okay, it's not overlapping. It's non-ambiguous. Every nucleotide is part of 1 codon read only once. Right, so it doesn't, the second time through, it doesn't read this set, right? The set of triplets is always set up by the start codon. It's almost universal. Almost all organisms, the codons have the same amino acid meaning. There's a few outliers out there, which there's a table at the end of this lecture or the next that talks about those. Again, being degenerate. Of all 20 amino acids, 18 of them are encoded by 2 or more codons. Each code, or each mRNA has a start and a stop signal. Where should translation start? Where should translation stop? AUG is the usual start signal for everything except very weird stuff, right? And defines the open reading frame. The open reading frame is really just this 3 by 3 by 3. So wherever that AUG sits in your mRNA is where translation begins. Towards the 5' end, never, ever, ever the very first 3 base pairs. There's always some other nucleotide sitting in front of that first AUG. And then it just reads 3 by 3 by 3 by 3. We'll talk about that again when we get to translation. The stop signals, or the stop codons, have no corresponding tRNA. No tRNA ever goes in and sits on a stop codon. It's actually a protein that sits there and blows the crap out of the ribosome so translation stops. Not a special tRNA. Do not pick that answer on a quiz or an exam. It's not a special tRNA that sits on a stop codon. It's a protein. No tRNA, no amino acid, none of that. Okay? The 3 stop codons are UGA, UAG, and UAA, listed right here. So let's take a looky-look at the genetic code chart. I know, it's pretty awesome. And what's even better is you're going to get this chart. I'm not going to make you memorize it because that would just be stupid. Who cares? If you end up studying RNAs and translation and so forth in graduate school when you're working on your PhD, you'll probably end up knowing them just from looking at it so much. But we're not memorizing it. What are we memorizing? Well, if we're smart, and we are, we're going to remember that AUG is always the start. Okay, it'll always be on this chart, but just remember, it's always the start codon. That's what you're going to look for in all of these little fun games. Right, here's our little stops. Up here, those will always be listed as a stop. The start is not listed as your start. It just says methionine, but we know it's the start codon, so we don't have to be told that. So what else do we know? This chart, again, will help point out the rules of the genetic code. So let's point out the whole degeneracy of the genetic code by using the chart. You're always going to have this chart when you come to that question on the exam. Presumably on a quiz, you'll have the chart, although the figures never show. Oh, I'll get to that in a minute. Let's not worry about that right now. So you can look at the chart and say, look, if we're going to do an example, one good example would be leucine. Okay, here he is, right here. How many codons code for leucine? 1, 2, 3, 4, 5, and 6. Anytime a ribosome reads one of those codons, the tRNA that's holding onto leucine is what's going to dock in that position and dump off leucine. Okay, each one of those. That means each amino acid, leucine, can be coded for by more than one codon. Can it? Yes, it's coded for by 6. Is 6 more than 1? Yes, it is. Woohoo! But what about each individual codon? Check the chart. Do you see UUA any place else on this chart coding for anything other than leucine? Keep looking. Do you see it? Do you see it? I don't see it. I only see it in one place, and that one place is right there. Each codon only codes for one amino acid. Each amino acid, on the other hand, can be coded for by multiple codons. Yeah? Yes? I like what you've done here? Yes. Okay, so that's a genetic code chart. You need to know how to read it because we're going to play the games of here's your RNA, right? Starts with a couple nucleotides, blah, 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 nucleotide, whatever. AUG sets up your reading frame. If the next 3 nucleotides are AAU, what the hell amino acid would come next after methionine? Let's find AAU. Here's the first base is A, which means it has to be somewhere in here. Here's the second base is A, means it has to come down here. Here's the third base is U, so it has to come over here. AAU codes for this guy right there. So Be sure you know how to use the chart because you will need to apply what you've learned, not just answer multiple choice about what does the degeneracy of the code mean. Yes, I know it's scary, but you could do it. I know you can. All right, let's go on and look at the experiments that they did to try to figure out how they figured out, well, what codons actually code for which amino acids. So let's look. So what they did was they made some homopolymers. All that means is a whole bunch of the same nucleotide all together. So that subverts the need to know how does it start, where does it track, how does it set up the triplet code. We know it's a triplet because it doesn't matter if the triplet is here or if the triplet started here because it's going to be U no matter what. Okay? And so they radioactively label an amino acid, one of them, and the other 19 are not labeled. So again, radioactivity allows us to track which one is the one we're looking for. And so they randomly pick an amino acid, they radio label it, put it in there in the tube with the 19 unlabeled amino acids, right? Put their little polymer guy in there as well, and then incubate at 37 with all the necessary stuff. Right? We need the translation system, everything in there, ribosomes, ATP, all the things you need to do translation. We let translation happen. We isolate the protein, right? We can filter them through the, filter them through a filter, right? Where it's going to trap radioactivity, okay? Listen for what's radioactive. And then look to see which amino acid is stuck in the filter. Free amino acids that are not built into a protein are going to go through the filter, right? If you think about a filter, right, it has little holes. So here's that little piece of membrane, has little holes. Individual amino acids can go right through. Woohoo! But a whole chain of amino acids are not going to fit, right, through that hole. The only one that's going to be in a chain are the ones that got incorporated into the translated peptide, which meant all the U's, U. That's the amino acid that was docked in the position that U coded for, right? So these guys are all hot, hot, hot. Those will all be up here. Anything that's not radioactively labeled will come through if the radioactively labeled amino acid is the one that we want, right? And so then we just do this for every single one. There's one radioactive. In this case, the radioactive one was proline. Everything else not radioactive, or lysine, or arginine, so on and so on and so on. Phenylalanine was the tube that lit up as being radioactive, right, that was stuck on the filter when they added the U, which means U codes for phenylalanine. And so they did this with all different types of homopolymers to see which one got stuck up on the filter. That's the one that it coded for, right? They had to do other experiments to look at all the other different codons, but this was one of the ways that they figured it out. Okay, so then they had to figure out, well, how do those amino acids get in there? How does that tRNA thing work? And so with the use of tRNAs, what matches between codon and amino acid could be determined. So again, they made very short mRNAs, essentially 3 base pairs, right? 1 codon. A whole bunch of tRNAs with all different amino acids, use a ribosome, mix all of this together, and see what gets built. Okay? So if GUU codes for, right, CAA has to be complementary, and that's the guy holding valine, that's the one that's going to be stuck to this ribosome. You could separate the free guys from the guys stuck on a ribosome, again by size, right? And that would tell us that GUU codes for valine. And they could do this with every single codon and figure out for each codon what amino acid does that code for. So same idea, they use the filter. The big stuff that's stuck to the ribosome tRNAs are going to be stuck up here. Sorry, that's my ribosome with a tRNA, right? With whatever amino acid. All the free amino acids, or sorry, the tRNAs with their amino acids are going to come through. Depending on which codon you stuck in there, tells you which one sticks to the filter, and you can figure out all of the codons using this method. So that's the experimental method for how they figured out which codon in the mRNA codes for what amino acid. And that's how they also figured out what tRNAs are involved. And we'll get into the tRNA problem when we do translation. But for now, thanks for listening.
Would the doctor care for a brandy before retiring?
No. Thank you.
Some warm milk? Perhaps?
No, thank you very much. No thanks.
Ovaltine?
Nothing. Thank you.